We're continuing our lecture on environmental factors which influence stomatal opening and closing. So I'm going to jump in and talk about CO2 concentration. This is a photograph of a free air CO2 enrichment experiment, or FACE experiment. This is at Duke Forest in Loblolly Pine, where without chambers they expose, in this case forest, to elevated CO2. And in those types of experiments and other experiments, we found that stomata tend to close a little bit as CO2 rises. You can think of that response. Remember, I said you could categorize responses in two broad ways. First, if there's a factor that influences photosynthesis, that will tend to cause stomatal opening. So for instance, if the CO2 concentration inside the leaf goes down, then the stomata will tend to open in response to that, and that allows more CO2 to diffuse in, and that allows photosynthesis to occur. So elevated, sort of the converse of that, is the elevated CO2 concentration tends to cause reductions in stomatal conductance, or increases in stomatal resistance. The thinking is that this might, as atmospheric CO2 continues to rise, this could result in increased water use efficiency as CO2 rises. In other words, water use efficiency is just CO2 fixed per unit H2O transpired. So that as CO2 is elevated, photosynthesis rate might be increased or maintained while less water is lost, and that could cause increased water use efficiency. These elevated CO2 experiments have shown mixed results with regard to that water use efficiency piece. Part of that is because that's a complex response. Part of it might be some acclimation going on. It's hard to tell, but what does remain is that in general, elevated CO2 tends to cause some reductions in stomatal conductance or increased stomatal resistance. In terms of response to leaf water potential, stomatal resistance tends to have a threshold response. So this is an example here of a threshold response where over some range of water potential, there's no change in stomatal resistance. But at some threshold, in this case at approximately minus 1.5 megapascals, stomata start closing. So stomatal resistance starts increasing relatively rapidly. That's what I would call a threshold response. And that tends to be what the response of stomatal resistance is to leaf water potential. Here's some data that show that response. Here, this is stomatal conductance. So we're looking at the inverse here, the response of stomatal conductance to leaf water potential. And you see these are negative megapascals, so these are negative values. -1, -2, -3. So again, over some range of water potential, in this case down to about minus -1.5 megapascals, there's no response of stomatal conductance. But at about minus 1.5 in this particular species, this is jack pine, we start seeing stomatal closure, and stomatal conductance declines as water potential becomes more negative, until at, in this case, about minus 2.5 megapascals, stomatal conductance is about 0. It's the stomata are completely closed. This threshold response, keep this in mind because this becomes important when we talk about the hydraulics of xylem and how that influences stomatal dynamics. So we'll come back to this in a later lecture. Stomatal resistance or stomatal conductance also responds to hormones, often generated in the roots. And this was demonstrated in some clever experiments that used split pot setup. And the way this worked is shown in this diagram, where we have a plant here, and the root system's been split and placed into two separate pots. So it's been trained to grow into two separate pots. And in the control treatment, which in the figure in your diagram is the filled circles, both pots are well watered. So both sides of the root system are getting plenty of water. In contrast, there's another treatment where one pot receives sufficient water, and then the other pot is allowed to dry down. It's allowed to dry out. So the soil in that second half of the root system dries out. And so they monitor those two treatments over time to see how both water potential in the leaves and stomatal resistance changes. So here's what they found. So again, we've got the control treatments, the filled symbols, and the open symbols are the treated ones, where one half of the root system is allowed to dry out. And the treatment is initiated at day 0. So what we see for leaf water potential is that over time there's no difference in the two treatments between leaf water potential. What that's telling us is in the treated plants where half the root system is in water, those leaves are getting plenty of water and they're not being essentially adversely affected in terms of water potential, because they can get plenty of water from that well-watered side of the root system. On the other hand, in terms of stomatal resistance, what we see is that eventually that treatment where half the root system is allowed to dry out, we start seeing stomatal closure. So that stomatal closure is not in response to water potential, because the water potential in the two treatments is the same. What they found out is that this stomatal closure is associated with elevated hormones, in this case abscisic acid or ABA. So abscisic acid or ABA is elevated in the treated plants, and that causes stomatal closure. Okay? So that dried root system, that dried part of the root system, is generating ABA, and that's causing the stomata to close. That ABA response is a mechanism that allows plants to sense drying soil and to have stomatal closure even in the absence of a change in water potential in the shoot. This is, as we mentioned earlier, a potential signal for feed-forward response of stomata. So that hormonal or ABA response is one mechanism by which that feed-forward response can happen. So just to summarize, we went over a number of environmental factors that can affect stomatal conductance or stomatal resistance. So light causes stomatal opening. And recall that there's both an indirect and a direct response to light. Air humidity, so drier air tends to cause stomatal closure. CO2, increased CO2 concentration tends to cause stomatal closure. And maybe it makes more sense to think of it the other way, and this is part of that indirect response, is that decreased CO2 causes opening, right? And then decreased water potential causes stomatal closure after some threshold. So it's a threshold response. And then finally, as we just talked, abscisic acid produced in roots can cause stomatal closure. Now, you know, there's one factor left that we didn't talk about, and that's temperature. It seems like temperature might be important for stomatal responses, but it's not. Studies generally haven't shown a direct response of temperature on stomatal conductance. There may be some situations where temperature interacts with other factors, and certainly temperature, for instance, increased air temperature can cause changes in air humidity that drive stomatal responses, but there's really no direct response of temperature, of stomata to temperature. So that's sort of an interesting thing. Stomatal conductance is one factor that's not heavily influenced by temperature, unlike many other physiological processes. Coming back to the bigger picture, recall that there's really three factors involved in leaf resistance. And we've spent a lot of time talking about that stomatal resistance part. But again, there's two other resistances associated with leaf resistance. One of them is boundary layer resistance. Boundary layer resistance is, boundary layer resistance is associated with a layer of laminar air near the leaf surface. Now, in just the atmosphere, air tends to be turbulent. So away from surfaces, the air is sort of moving randomly in different directions. But as that turbulent air encounters a surface like a leaf, it slows down and it becomes more organized. And that organization and slowing of air causes that turbulent air to become laminar. That laminar layer is sometimes called the boundary layer. Okay? And that boundary layer is associated with boundary layer resistance. And that's because in turbulent air, water molecules are rapidly transported. So in turbulent air, water molecules move easily. But in that laminar sublayer, that boundary layer, the movement of water molecules away from the leaf is slowed somewhat, and actually that air or that water vapor has to move by diffusion. Diffusion is much slower than turbulent transport. So the boundary layer can act as a resistance to water movement away from a leaf. So this boundary layer can resist, act as, or it can be a resistance to water transport. The thickness of the boundary layer, so boundary layer resistance, is proportional to the thickness of the boundary layer, and that's affected by two things: leaf dimensions, where larger leaves have thicker boundary layers. And that's just because on larger leaves, there's a greater distance over which friction takes effect, and that allows a thicker boundary layer to build up. So all other things being equal, larger leaves tend to have greater boundary layer resistance. And then second is wind speed. Slower wind speeds lead to thicker boundary layers. So sort of the maximum boundary layer resistance would occur in large leaves at low wind speeds. That's when boundary layer resistance would be greatest. In general, boundary layer resistance is much less than stomatal resistance. So for most leaves in most situations, this boundary layer resistance is a lot smaller than stomatal resistance, which means it's not an important impediment to water loss. So usually boundary layer resistance is not an important factor for controlling water loss. Now with really large leaves, under really low wind speeds, that may be a situation where the boundary layer resistance actually becomes the major resistance to water loss. But that's sort of a special case. Where the boundary layer resistance becomes more important is for energy balance. So when we talk about energy balance later, boundary layer resistance will come into play again. Okay, so we've talked about stomatal resistance a lot and boundary layer resistance. The final resistance is cuticular resistance. Cuticular resistance is just the resistance to water vapor diffusion through the epidermis and the cuticle. Remember, the cuticle is that waxy layer on the surface of the leaf, and in general, this is very large. In fact, usually, it can be assumed to be infinite. In other words, under most cases, we can assume that the cuticle is impermeable to water vapor. Where cuticular resistance becomes important is when the stomata are completely closed. So under really extreme drought conditions, when the stomata are completely closed, we can measure cuticular resistance and find that the cuticle actually does allow a little bit of water vapor diffusion. There's almost no cuticle that's completely impermeable to water vapor. And you might actually see some species differences in cuticular resistance. So when stomata are completely closed, under extreme water stress, we may at that point detect species differences in cuticular resistance, really just associated usually with a thicker cuticle. And people have measured thickness of the cuticle basically by dissolving the cuticle off of leaves and weighing it in different species and comparing those. So under extreme drought conditions, differences in cuticular resistance could make a difference in, for instance, survival.